All right, let's get started. It's a great pleasure to introduce to you Mike Paulin, who's a longtime friend of mine and has done really, really interesting work at the intersection of neuroscience and mathematics for a long time. Mike's right now, it's 5 a.m. tomorrow for Mike, so he's
That's true.
Literally a thinker ahead of his time.
Going to be awake real soon.
And he's gotten very little sleep since coming from New Zealand, but I think you're going to find his talk really fascinating. This is very dear to my heart, this topic, and I just can't wait to hear what he has to say. So, Mike.
Okay, thanks, Malcolm. And thanks for inviting me. I'm on my way to Virginia, and when you're in New Zealand, Chicago kind of looks like it's on the way to Virginia. So here I am. So I'll just start off by getting you oriented in time. So here's the history of the Earth. And pretty much as soon as rocks cooled, there were bacteria. And not long after that, these really complex organized communities of bacteria inhabited the world for a billion years or so, till the eukaryote cells turned up a couple of billion years ago. And eventually these, we've gone from pond scum to these highly intelligent, agile, conscious creatures, and that's the question that interests me, is how do we get there? How do we get from this stuff to that stuff? The interesting stuff happens clearly right around the Cambrian explosion, around more than 500 million years ago, and it's now known that there was a fairly widespread community of animals living long before the Cambrian explosion. So 90 million years before the Cambrian explosion, we now know there were lots of soft-bodied animals and complex ecosystems. This is to orient you properly in space. This is a map produced by New Zealand's official wizard. That's where I'm from, Dunedin in New Zealand. Queenstown is a couple hours drive away. You should come there next year. We've got the Australasian Winter Brain Research Conference in the last week of August, the Australian Neuroengineering Conference, and only half-day sessions. Near ski field's only 20 minutes away. When you've finished there, you might want to go somewhere a little warmer, and I can recommend going to Australia, a place called Ediacaran, about 600 kilometers north of Adelaide. And the Flinders Ranges, I actually went there a long time ago, and I put that slide up before, so you realize that this is just the other day, really. I mean, you'll find out this is quite recent. But back in the day when New Zealand was a socialist paradise, and if you passed your exams, you not only got to go to university for free, the government paid you enough that you could just hang out for the summer. In fact, they paid you so much that you could go to Australia and hang out for the summer. So me and my girlfriend went and kind of hung out in the outback of Australia and spent some time camping in the Flinders Ranges. And we went back there in January, and we get a new perspective on it, because what we didn't know then, and it's pretty clear when you look at it, is the Flinders Ranges are actually an ancient seafloor that has been tilted up on its side and sticking up out of the desert. So the centre of Australia used to be an ancient shallow ocean until about 500 million years ago, and the Flinders Range is the bottom of that ancient ocean tilted up and sticking into the desert. And it's when you know, why does that always happen? So it's kind of a no-brainer when you realize it's an ancient strata. This is an ancient seafloor, right? So this is an ancient seafloor, and if you go there, there's a magic bus that will take you through this ancient seafloor. And if you go there in January, you'll be the only people there, because it's 115 degrees in the shade and there's no shade. And the only people that are crazy enough to go there are a couple of Kiwis, so that's what we did. Here it is from space looking down on the Flinders Ranges, and so here's this ancient seafloor which has been buckled and broken, and the edge is sticking up into the desert. And if I use my little machine here, this one here is that Brachina gorge that we looked at, that we were looking at before. And so if you head up the main road from Wilpena to Blindman, you head off into the Brachina gorge. You go a few kilometers up that road, and there are fields of stromatolites, right? These rocks, and they're quite interesting, but you can come and look at photos later. You go another couple of kilometers up the road, and here is this brass pin in the side of the cliff, and this is the official marker of the start of the Ediacaran era, which was just created in 2004. So it's a new geological era that's been created now that it's understood that there were animals for a long time before the Cambrian explosion, this group of animals called the Vandians or the Ediacaran fauna that are found globally all over the planet and now in about 30 different locations. These same ecosystems exist, and this is the official marker for the start of that period. And that is the strata that you drive into as you enter the Brachina gorge, the boundary of the Ediacaran. You go 16 kilometers up the road, it's about 10 miles up the road, and you hit the boundary of the Cambrian. So you're driving through the Ediacaran period. And so I kid you not, we went past this sign that said Cambrian, Precambrian boundary, and the driver just carried on. And I said, hey, can I stop, you know? And he said, there's nothing to see there. And I said, yeah, but I stop and I get a photo, right? So we stopped and I took, Helen took the photo. And then he said, well, there's this little gully up here, and I was with this geologist, but it's just some wormholes. And so he went back to the van, and I went up into this little oven. And sure enough, there, right on that Cambrian boundary, are the burrows of worms going into this ancient sediment. Right? And you go another couple of kilometers, and you start to see these complex animal fossils, skeleton shells, all that stuff. Right? So you literally drive in the morning through the Ediacaran period. In the middle, there are some fossils to see, and if you go with a guide, they can take you and show you some broken and busted fossils. But all the good stuff has been taken to the museum, because when this place became known, people would head up there with their pickup trucks and dynamite and go looking for these things. So now the good stuff is taken out, but here is an animal from the Ediacaran, something like a sea pen, looks like a plant, but it turns out that it's something like a sea pen, a filter feeding organism. Dickinsonia was the supervisor of the student who first discovered these things and whose papers got rejected by the Geological Society because they said there's no animals before the Cambrian, so seriously, why are you sending this stuff? And he didn't get his master's degree. So Dickinson was his supervisor, and I think it's appropriate now that the first known animal that appears to have a head is called Sprigina, after Reg Sprigg, who was the discoverer. So these fossils are all over the place if you know where to look for them in the Ediacaran period. Now, we are fairly confident that these animals had no kind of internal organs or nervous systems, and if you just have a little trouble getting your head around the idea that there are animals with no there could be animals with no nervous systems, there are two groups of animals around on Earth today that do not have nervous systems. And one are the sponges, the poriferans, and the others the placozoans, these little amoeboid-like creatures. And the interesting thing about the sponges, although they lack nervous systems, they don't lack movement, they don't lack adaptive goal-directed behaviors, they just don't have any spiking neurons, they don't have any nervous system. Right? So some sponges can actually crawl around. They can cough. If they get silted up, they can cough. It's kind of slow, but they can cough. And these guys, their larvae, can swim around, and they swim around clearly in a goal-directed way. These things are clearly capable of following gradients of sensory cues and finding environments in which to settle. So these animals are actually quite fascinating, have interesting behaviors, feeding behaviors, but no neurons, no nervous system. They are brainless animals. I put this slide up because this field is in kind of a state of flux now. The reason I got to this point was when I first met up with Malcolm in California years ago, I was working on sharks, and I did shark sensory systems for my PhD, and the reasoning was, well, human brains are too complicated. We should find something really simple, like a shark, and we'll figure that out, and then when we figured that out, we can figure out human brains. And by the time, a decade or two later, when I met Malcolm, I had realized that sharks are just too complicated. The shark nervous system is just, it's so complicated that you just go backwards trying to figure out what's going on there. And so it was actually Malcolm who introduced me to this theory about the evolution of the first animals with neurons due to Nielsen, the trochea theory, the idea that it was these sponge larvae that instead of settling onto the sea floor and becoming sessile filter feeders, stayed in the water column and became active feeders in the water column, and that was the path that led to animals with nervous systems. And I still really like this theory. I mean, it's kind of a no-brainer, isn't it? Getting from here to here, that really appeals to me. But I have to say, the more I read about this topic, the less clear it becomes to me what the pathway was, and all kinds of things are possible. It's not clear whether our ancestors were in the water column, whether they settled to the floor. This was a hypothetical animal proposed by Haeckel in the 19th century, and it's pretty clear that what he drew essentially was this thing, right? There are these animals, so now we know about all of these complex animals in the Ediacaran. I think it becomes really suddenly quite unclear what the pathway is. However, what we can figure out is roughly when neurons first appear, and by looking at fossil record, looking at molecular biology, looking at phylogeny, try and figure out where on that time scale did neurons turn up, or where's the first evidence for neurons. And what we find when we do that is, and I won't go through the details, there's a paper coming out soon. Right after the Cambrian explosion, all these animals appeared. All the kinds of animals that we recognize today, pretty much, appeared very soon after this 543 million years ago. So most of the stuff we recognize today turns up here. Most of these things we don't recognize. We don't know what they are. But one thing is clear is that they're all soft-bodied. They're all either filter feeders feeding on plankton in the water column or on the seafloor, or they're grazers on those bacterial mats. So they are, and when you find the fossils, they're always intact. You never find one of these fossils broken up or bitten, right? These guys you often find in pieces. Often you find them in pieces at the bottom of a burrow, right? And so it's clear that predation, animals eating other animals, happened around this time. And the clue for us is that this weaponry that evolved at this time has evolved from multiple different biochemical pathways, right? So the different kinds of armor that animals developed at that time, using different genetic mechanisms to produce armor, suggests that there's some external cause. It's not a change in ocean chemistry that leads to skeletons and shells, as some have suggested. It's a change in ecology. There's something happening to these ecosystems that means one way or another you've got to start wearing armor, right? So we think there's strong circumstantial evidence that the origin of neurons, and again you can look at the behavioral evidence, the trace fossils and the burrowing. All of the evidence up until about 550 million years ago is consistent with the kind of animals that don't need neurons, right? That are just following gradients of things. They are doing simple goal-directed behavior. There's nothing that indicates there was anything out there that needed neurons. At about this age, 550 million years ago, you start to see evidence of both fossil trace evidence for behaviors that would seem to require neurons and fossil evidence of animals that are clearly in classes that we know today have neurons. So right around that time is when neurons appeared. So the evolution of neurons is associated with this transition from this essentially two-layered ecosystem where the animals are all suspension feeders and grazers to another trophic level in your ecosystem where now you have animals eating animals. And neurons turned up around this time. And what we think is the key thing going on here is that if your prey is very small, has a very low Reynolds number, it's not worth chasing individuals, right? You cannot get the, so if you are small, or what you're pursuing is small, it's not worth pursuing them individually. The energetics make it, I mean, even if you knew where it was, it wouldn't be worth chasing. So that means that information about where things are doesn't matter to these guys. So information about a concentration of organisms or where organisms are likely to be found, that kind of information is valuable, right? So these organisms all have sensory systems and they all have the ability to respond, as plants do, to gradients in the environment and move and adapt and move towards those gradients. When carnivory turns up, the situation suddenly changes. So now you're getting episodic encounters with rare and valuable food items that are definitely worth knowing about and definitely worth chasing, right? So that means that there's an affordance. It's now worth paying for information about where other animals are in your vicinity. Okay, so that's the kind of background story. Now if we look at predator-prey interactions, there are theoretical arguments and empirical data is clear and consistent. So this is just an example from a recent paper. What happens when, for example, two fish are near each other. This graph is from a zebrafish eating larval zebrafish. So this is zebrafish eating their own babies, basically. And the rule seems to be that the larvae have essentially, this is the probability of escaping, no chance of escaping if they don't try and make an escape maneuver before the predator strikes. And then as the distance increases, there's this steep S-shaped curve out to a point where they have a very good chance of escaping if they're at that distance when the predator strikes. So there's this very sharp cutoff. And the energetics of the situation, if you've ever been diving, you'll see this with the reef fish. The reef fish will just move very slowly. Right? It's very efficient to move very slowly in water. You don't want to waste any energy unless you have some reason to think that there is some imminent threat, at which point it's a good idea to move quickly. Right? So the energetics favor short bursts of activity. So if we look at this from an individual's point of view, if there's any chance that you're going to get eaten, dying is, it's kind of a bad thing. And so you might put a number less than infinity. I don't know what number you might pick, but for the sake of argument, we'll say that if you're in this zone, it really doesn't matter what you do, right? It's expensive for you to be in that zone. You don't want to be there. Out here, it's actually going to cost you something to flee, right? So there's some energetic expense of getting out of the way. And actually there's a benefit to staying as well, because you would be doing something like eating or something fun that you like to do. And so there's a benefit to not moving and there's a cost to moving. And so this would seem to suggest that what you need is some kind of threshold trigger. You need something that can, if you're this guy, if you have sensory systems that are able to detect something produced by this guy, you might want to have some kind of threshold trigger that would tell you to get out of here. You need some mechanism that is going to generate an escape behavior out here somewhere. Now, that becomes a problem because it seems like really you want to spend a lot of your time fleeing, even if there's a tiny chance that there's something out there, right? Because the cost of not fleeing when there's a predator is infinite, right? And the cost of fleeing when there isn't a predator is finite. And so you would think it's a good idea to spend a lot of time just kind of jerking around and wasting a lot of energy. However, if you look at this from an ecological perspective, this is the bad news, right? Is that from an ecological evolutionary point of view, the evolution doesn't really care about you, right? It cares about what happens to your lineage, and the bad news is that other things being equal, and this is true for all of us, your lineage is likely to go extinct quite soon, right? No matter what you do. And so over evolutionary time, dying today doesn't really matter very much, right? As compared to dying sometime in the future. It's not, over evolutionary time, this is really not very important. And however, the problem is that in this game, you are not only competing against this guy, you are competing against this guy. And that's what changes the game. So it's like, you know, the old joke of the hunter on safari, and he's aiming at the charging lion and the gun jams. So he drops the gun and says to the guide, "What should I do?" And the guide says, "Run!" And the hunter says, "Are you crazy? I can't outrun a lion." And the guide says, "I don't have to outrun a lion. I only have to outrun you." Right? And so that's the deal, is it's true that other things being equal, if you're all equal, the probability, what happens in populations of cloned organisms, is in a relatively short time, over a few thousand generations, all the descendants, all the members of the population end up being descendants of one member of that original group, right? And this is just statistics. If you start replicating and taking things out at random, no selection going on, just picking these things at random, everyone is a clone of one individual. And so other things being equal, if there are n individuals in your population, you've got a 1 over n chance that your lineage is going to be perpetuated in the population. So you really don't have much to lose because you don't have much of a chance to begin with. However, if you can reduce the cost of escaping somehow, so if you can increase the probability of escaping when there is a threat and not escaping when there isn't a threat, by essentially an arbitrary small amount, and you run the simulations and you can run this down to machine precision, right? A factor of 1 part in 10 to the 16 is enough to make a really significant difference to the probability of a clade surviving. If you can make a tiny difference in your feeding efficiency or your ability to tell whether or not there's a predator about to strike, then you become quickly almost certain to be the one that survives. And so there suddenly becomes, the cost is not just infinite for being here, the cost is infinite here, except for any guy that can figure out a slightly more efficient way of deciding when to escape. So that decision about when to escape is crucial. Right, so what marine animals today use is hair cells. So this is just a micrograph of hair cells. So fish and other marine animals are using hair cells either to detect water displacement or electric fields. And those things tend to have some kind of a dipole field structure around a dipole water movement field around a moving object in water, or a dipole electric field around some animal in water. So animals have evolved these sensory systems that are able to detect those dipole fields, and physics really comes to your aid here because the amplitude of that stimulus from a dipole field is dropping with the cube of the distance. So you get a stimulus which is very small at long distances and increases very rapidly at close distances. And so then creating a threshold trigger that's going to get you to escape at the right point is just a matter of setting the sense of having something that can detect that dipole field and setting the threshold correctly. And that, just because of the physics, is going to give you a high probability of escaping when there's a real threat and a low probability of deciding that there's a threat when there isn't really one present. So the physics is your friend in that situation. However, as you drive these things out to further and further distances, because of noise in the environment, these things are going to get unreliable, right? So what you're really going to get is maybe at the beginning, it's when things are moving really slowly, it's worth knowing that there's something right here that's about to eat you. But after a few million years, you need to know if there's something out here that's going to eat you. Right now you're at some distance, and you need your senses to be able to detect that at a distance. What's going to happen, again, because of the physics, is you can't have a threshold trigger anymore, but you can have something where the probability of this thing triggering is determined by that stimulus from the predator. So you're going to end up with some stochastic process where this thing is more likely to go off when there's a predator close and less likely to go off when there's a predator distant, but it's going to be going off all the time, right? This is still going to work. It's still going to work because it has that property of being more likely to generate an escape behavior when there is something close that's likely to eat you. And there's been, again, I think it's easy to imagine that this is true, but you can do the simulations and show that if you just create a model with a bunch of creatures which have senses of this kind, and you just run these evolutionary simulations where these things reproduce and they have a tendency for their thresholds of their descendants, the sensitivity of the detectors in the descendants to be similar to the sensitivity of the detectors in the parents, then you very quickly get these things selected for. They all turn out to have the same sensitivity. They're all sitting at that same point. So this gives you a way of escaping from predators, but it's kind of inefficient because you're actually generating these escape behaviors a lot of times when you don't really need to, right? You've got this random process going on. It's happening at a low rate, but you're still paying a price. And if somebody can figure out how to not pay that price, that's worth infinity in evolutionary terms. If someone can figure out a way that instead of responding every time one of your sensory neurons fire, you could figure out when exactly should I escape. Given what that sensory neuron is doing, when do I have to make my break? And so then we can look at it from a Bayesian inference point of view. We can say, I have some data. My data is this spike train, a stochastic process. I have the sensor which is firing randomly, and I want to make a decision about, by looking at that stochastic process, is it likely that there is a predator about to pounce on me given that pattern of sensory input that I'm seeing? And the answer is now, so if we characterize the sensory input as a stochastic process, so we have given this state variable, which is the distance to the predator, there is some probability that the neuron with some firing rate, right? So there's some parameter of a stochastic process that depends on this variable that I'm interested in, right? And I get samples from that stochastic process, right? And those samples from that stochastic process are the spikes. They are samples from, in the limit, a Poisson process. And the parameter of that Poisson process is going to tell me how far away the predator is if I could tell what that parameter is. Right? So what you need to do, given some observations from a stochastic process conditional on some state of the world, you can just use Bayes' rule, right? Or I can just use Bayes' rule, right? I got MATLAB. I type in Bayes' rule, and that gives me the probability distribution for what's true in the world given this pattern of spikes that I've seen coming in from my senses. And it turns out that the critical rule, as you might imagine, you want to know, is it likely that there is something in that critical zone? And it turns out the Bayes optimal decision is that this guy should flee when Bayes' rule says that the maximum of the conditional probability density of prey location, given the spike train that I've seen, is at the boundary of the death zone, right? And that means if any of these guys could do that, if there was a way that these guys could actually solve Bayes' rule, that, right, so we're looking here, here's the death zone boundary, so if you kind of think about it in terms of this picture, where is this guy escaping? If your system is causing you to escape out here, which is the kind of safe thing to do because it's really dangerous to get close to this boundary, as long as there's any space left, another guy can come in behind you, right? If it could do Bayesian inference, it could come in behind you and be better. It could outperform you. So the answer is, if it is possible for somehow for an animal to infer from a Poisson spike train some parameter of that, the parameter of that spike train, then that creature could outcompete all of its conspecifics, and it would almost certainly be the one that survived. Right? That decision rule is necessary if it's possible. So we look at how you do Bayesian inference from spikes. So this is just Bayes' rule. So we're working out at a given time, we're trying to estimate the parameter of a Poisson process. That parameter is a function of the variable that I'm interested in this case the distance to the predator at some time. Depends on the prior probability density, right? So if I'm, so this is spike by spike, right? As a spike comes in, before that spike arrived, I had some prior probability distribution for the distance to the predator. When the spike comes in, I can update my belief about where the predator is. And the rule is, this is the likelihood function. So this is the likelihood function for a Poisson process with a refractory period, right? So for a Poisson process with a small refractory period, this is just Bayes' rule. And you don't have to believe me. Here's I'll just show you a simulation. So I'm just here solving Bayes' rule. And I'll run this through a couple of times and so you can watch it and figure out what's happening. So here is a Poisson spike train with a fixed parameter. And the fixed parameter is, in this case, 0.5. So here we are looking at the parameter. There's my estimates of the parameter. Here we're looking at the distance to the target. Right? So the parameter of the spike train is a function of the distance to target, so I can just do the transformation between these probability distributions. What we have is the blue plots on the right are the posterior probability distributions given this particular sample. So this is one sample taken from this stochastic process, right? So the sense organ is generating these observations from this process. Here's a particular observation from the process. The blue functions out here are the posterior probability distributions given that spike train. And the purpley ones are the expected. So that's what, on average, we would expect the posterior to be if you observed a process like this. Here I'm plotting the 95 percentiles of that distribution. Here's the true parameter in green. And here's how this is the expected lower 95th credibility interval, the upper 95th credibility interval, and this is the maximum posterior probability expected. And the blue line here is the maximum posterior probability for prey location, given that spike train. And at the circle points there, those are the most likely location of the predator at the instant that that spike is observed. And so we run it through again, and you can watch what happens. And so what happens is you see that in the interspike intervals, when there's no spike, the posterior density is drifting away, so that when there's no spikes, you're getting information that's telling you there probably isn't something close. And when there's a spike, that tells you maybe there is something close. And so you'll see those distributions. You'll see particularly if you watch this one, the prior distribution is exponential in lambda, so this creature is assuming to begin with that there isn't anything close. So it starts out saying, I don't think there's anything about to attack me right now. So the prior distribution is over here, and you'll see this posterior distribution jumping towards the true location as those spikes come in. So as Bayes' rule tells us where the target is likely to be. Right? So you see this distribution jumping every time there's a spike. It jumps that way. And in the interspike intervals, it floats back out. I'm safe. No, I'm not. Right? And so this distribution, if you leave it long enough, will converge to a delta function, right? Saying this is where the thing is. That's just the central limit theorem kicking in, right? And I've put actually it doesn't matter actually what the relationship is, as long as there's some relationship between what you're interested in the world and the parameter of the process that you're observing. It doesn't matter what that relationship is, you just put it into Bayes' rule and it tells you what the posterior density is. So this tells us that this is possible if you could do Bayesian inference with neurons, with spiking neurons, you would win, right? You would take over the world if you could figure out how to do that. Here I've got just some snapshots of the previous simulation. I've plotted out the maximum posterior probability for the spike train, and here in this case, here's this one's easiest to look at. The green is the true location of the predator, the true distance to the predator. The red is the critical distance, right? And so this creature has to decide whether or not the signal it's looking at is coming from something that's closer than this. And if it can figure it out using Bayes' rule, it will be the one that survives. And here I've just plotted the maximum posterior probability for the spike train. And the point where that crosses that threshold, the point where the maximum point of that distribution crosses that critical line, is the point where it has to make the decision to go. And if it makes that decision, nobody can outcompete, right? Nobody can beat that. And I actually came into this backwards, but I'm going to play it forwards. Because if you look at this, you think, well, gee, that kind of looks like the membrane potential of a neuron, doesn't it? If you send a spike train into a leaky integrate and fire neuron, doesn't that kind of look like the membrane potential of a leaky integrate and fire neuron? So I thought, well, hey, let's just check it and see what happens. Can I actually predict this threshold crossing point with a leaky integrate and fire neuron? So I've got my sensor, which is a spiking neuron, and it's spiking because the laws of physics and the laws of evolution say that's what you have to have. It's a Poisson process. It looks like you could put a single neuron in behind that sensor, and it would tell you when that threshold crossing point is. Now that turns out to be true, but in a slightly surprising way. Because when you do the simulations, you discover absolutely, indeed it's true, that you can get a single leaky integrate and fire neuron to trigger on every trial exactly at the point where that maximum probability crosses the critical distance. But it doesn't actually look like the Bayesian maximum posterior probability. So this is a weird thing that I actually haven't figured out yet. You can get a neuron, you set your critical distance, you find a neuron that crosses the threshold at that point, and that will work for arbitrary samples from that distribution. So now I can take arbitrary spike trains and feed it into this neuron, and the membrane potential of that neuron will hit that threshold exactly at the time that the Bayesian maximum posterior probability hits the critical point. on every trial, right? But it's not the same, right? So up here we're looking at, what are we looking at? Expected value of the maximum posterior probability. So so in this case, the target really is at distance 1. If the critical distance is 1, it actually takes forever to figure that out, right? So if the critical distance is, if the predator is actually at the critical distance, it's asymptotically, it's going to get there. But as soon as the predator comes inside that critical distance, this thing figures it out very rapidly. And I've only done this in simulation up to this point, but and I've been all over this parameter space, and it seems to be a very robust result that the time when Bayes' rule tells you you must escape if you want to survive is exactly the time that a single leaky integrate and fire neuron fires, right? So maybe this is a coincidence, but isn't it interesting that a single neuron, the membrane potential of a single neuron, turns out to provide the Bayes optimal solution for the problem that is arguably the problem that drove the evolution of nervous systems. So maybe that's a coincidence, but I think it's too much of a coincidence. And I know something about why this is true, because if you go back and look at Bayes' rule, you look at the likelihood function for a Poisson process. The likelihood function for a Poisson process is the impulse response of a first-order linear low-pass filter. It's the impulse response of a neural membrane, right? So it's to do with this much I know. Now you can ask the question, given that you have a leaky integrate and fire neuron that has the capacity to tell you whether or not there's a target at a particular distance, what if you had a whole population of these things? Maybe they could actually calculate the whole posterior density, because this solution, again, if you think about it, this solution is fine, it's optimal, but it's optimal only for about 5 minutes, right? Because as soon as it's only working for particular prey at particular distances. So as soon as your ecosystem gets any more complicated, this is not going to work anymore. And you really need to step this up. You need to estimate the whole posterior distribution so that you can figure out what to do. Now, it turns out that with a little bit of effort, it's absolutely true. You can get a whole array of these things, and they will exactly give you the whole posterior distribution, right? So you get sensory spike trained sensory data coming in, and a bunch of these leaky integrate and fire neurons. And here I am just mapping the distance that the neuron is tuned to, if you like, into space. So when a neuron, so you can think of these neurons as voting, saying, I think it's here, right? And another neuron saying, no, I think it's here. And it's the spatial distribution of the firing pattern in those neurons maps onto, in blue, is just the spatial distribution of these spikes, right? So that's just where the neurons are that are firing at any instant. And in magenta is calculated by Bayes' rule. So that tells you what the real answer is, the Bayesian, the 95% credibility interval and the maximum posterior probability from Bayes' rule in magenta. And this bunch of neurons is getting that exactly instantaneously. There's no lag. It's every instant this thing is exactly giving you the posterior density. And if we look at that in a static map, so here I am just looking at the marginal distribution here. Again in blue, looking at which neurons in this map are firing. In magenta, what's the Bayesian posterior probability density given that spike train at 10 seconds and 50 seconds and 90 seconds and 1,000 seconds. This thing is giving me exactly the posterior density. So that's where I'm at this point. I can figure out how to use spiking neurons to solve these simple state estimation tasks. And I think perhaps this was the task that neurons first evolved to do. Thank you.
Questions? Mike?
So I missed the last slide. How fast is this again? What's the horizontal scale?
So the time scale is arbitrary, so these are time units. But if they're seconds, this thing I think has a firing rate of half a spike per second, I think. Right? I think that's about right. So, I mean, if we call them seconds, you know what it's like in computer simulations, just right. So if you
shouldn't there be an interest in how quickly you can make this decision too?
it's instantaneous. It's instant. You get the Bayesian posterior density is all the information that you have at that instant, and this mechanism is instant. This convergence is because that's how fast the information is coming in. The computation is being done instantly. There's no lag.
But can it make an efficient decision instantly?
It makes an optimal decision at every instant, immediately. It is optimal, right? We go back to look at, say, this graph. At every instant, at every microsecond, up to this point, if it goes at that instant, it's going to get wiped out, right? The cost on an evolutionary timescale, the cost is infinite if it goes a microsecond too early. Let's go milliseconds because that's the real timescale of sloppiness in neurons, right? And so it is at every instant making exactly the optimal decision. It's saying no go, no go, no go, no go, bang, go. There's no delay. The only delay is in how much you're prepared to invest in your sensors to get the information. If you want to build more sensors, get more information quickly, you'll be able to make the decision quickly. But this rule, this decision, this is Bayes' rule. Given the information that you have instantly, this is the decision that you must make.
So the question is, how quickly can you accumulate the information?
That's what, yes. If you, and so it actually turns out when the real world contains a lot of uncertainty, you don't actually need to know the posterior density very accurately to make something very close to a Bayes optimal decision. Right? So if real uncertainty is high, which it is in natural ecosystems, you can actually do very, very close to optimal with quite a bad estimate of the posterior density. Right? And so,
along those lines, the limiting factor for the speed is the firing rate of the spike.
it's how much information is available about the variable that you're interested in. Given that information, this mechanism instantly tells you what decision you should make.
But if you increase the spiking rate, you would get better sampling.
But as I just pointed out, I could call this milliseconds. Now I mean, it's just a simulation.
Yeah.
Right? If I call this milliseconds, my creature's making a decision in 10 milliseconds.
No,
that's pretty fast.
So in principle, in a system like this, you would favor very rapidly firing sensory domains.
That's to the dynamics of the structure.
The energetics favors having an array of more slowly firing neurons. It's more efficient to have many neurons firing more slowly than having a few neurons firing rapidly.
So I'm not sure this really makes sense, but I guess I was expecting you to sort of suggest why spikes, why neurons. We went from these gradient-based animals, and I understand it at this level of description, but it seems to me that your Bayesian optimality has already got the notion of spike built into it.
Yeah. The reason for spikes is because of this thresholding property. In other words, there's a very sharp boundary in the state space, if you like. There's a region in the state space, and if the posterior probability, maximum posterior probability of the world state gets into that region, the costs are infinite in both directions, right? That's what drives it to be a spike. Because if you go too early or you go too late, the cost is infinite. You have to go exactly at the point, right? So that's what drives it to be a thresholding, spiking process. You have to make a decision at an instant. You talk about timing. I mean, everybody's about, the cerebellum is about timing. It's all about timing. It's not about timing. It's about state. You've got something coming at you, and there's a boundary in the state space. And when it gets across that boundary, you instantly have to go. So that's what makes it necessary to have a thresholding device. You have to have something that switches instantly when that state, instantly as soon as that condition occurs. So that's what drives you to have spikes. And it's the fact that you need to do this at increasing distances and the fact that there's noise in the world that means that, it's a story. I'm just telling a story, but the story seems plausible that as you drive these things to be more and more sensitive, you just drop into the thermal noise, and instead of having this thing that triggers when you cross a threshold, it's going to be going off all the time. It's just more likely to trigger as the target gets closer. And that then creates this opportunity to come in with another neuron and only trigger when you have to.
I had the same question as Mike, but what you just said to me is, if you think about the first predator-prey relationships, like Warhols and Claudina, you've got a prey which can't escape because it's encased in its skeleton, and that's the first empirical evidence of predation in the evolutionary record, that you would expect that the delta function there is actually graded initially because you've got slotty animals and animals that can't escape.
Yes.
But then you would expect graded potentials for your receptors, which is what you see. Photoreceptors are graded potential devices, right? But once you put an afferent on them, you have spikes. But the receptor itself is a graded potential.
Yeah. So the Claudina point is true. That's the first empirical evidence for predation. But I would argue that that's not true because Claudina has a shell, has an exoskeleton.
Right. It does have an exoskeleton.
But that's how we know. That's how we know that we're predators, because we see those exoskeletons with holes in them. But why did they have it? Exoskeletons are costly. If you've got to carry around this heavy armor, you're not going to do that. You're going to die if you carry armor that you don't need. Right? So if Claudina has already evolved armor before we see the drill holes in it, something must have been predating it. That would be my argument. So predation must have started.
Right, but I'm just saying, like, in the initial picture, it probably was graded sensors rather than
so
spikes.
You're exactly right that it's only in the limit as these things get bigger and bigger and faster that this function becomes steeper, and because the actual Bayes rule would say that you actually want to match your posterior. You want to maximize the posterior probability that the predator is outside this zone, and that it isn't a delta function if this thing isn't a cliff. Right? It only becomes a delta function as these animals get so fast and so big that this thing becomes the boundary becomes very steep. So you're absolutely right. Initially, it doesn't have to be the initial spikes could be big, long, sloppy things. But we've got millions of years to play with.
Yeah.
Let me try.
So if I've understood correctly, quite a bit of your argument hinges on the idea that it's not just predation that's driving this, it's the ability to outrun or outmaneuver your conspecific.
Yes.
So then, how do you adapt this to account for the appearance of collective behavior, which we see in very primitive organisms, both as a means of keeping that organism, that species safe, and as a mechanism of predation?
As soon as any change happens in an ecosystem, things can get very complicated very quickly. So this is not the only solution to this problem. One solution to this predation problem is to develop a shell, right? Become a mollusk. Then, another solution is to become poisonous. These are alternative solutions. Running away is not the only answer. Right? So you could develop a shell, you could become poisonous. They're costly. And there are all kinds of other, as you mentioned, other mechanisms that could evolve in response to predation. So this, I'm just arguing, is kind of circumstantial evidence that neurons. So I'd put neurons, and I don't think anyone's really thought about it this way before, but I'd put neurons in the same class as armor, literally. It's a response to predation, right? It's not that, I mean, nobody argues that animals develop shells in case predators turned up, right? It's never going to happen. After predators turn up, this is a good thing to do. It's worth paying. It's worth paying for a shell if there are predators around, right? And it's the same deal with neurons. So there are people who argue that, you know, some random mutation caused eyes to evolve or neurons to evolve, and this suddenly allowed creatures to see and chase each other and predate. Well, this can't happen. You can't have things evolving spontaneously unless there's some price that you're paying.
But I think what I'm missing is whether the statement you're making is that neurons evolved in response to predation,
Yeah.
or neurons evolved in response to predation and the need to outrun your conscious.
Well, that's just a consequence of predation. It's just if something's chasing, you know, if you're a member of a group and something's chasing the group, you just don't want to be the slowest guy in the group.
Unless you're working, but you said, as you said, the individual doesn't matter. So the way that it doesn't matter if you're the last one is if you're working to ensure the evolution of your species, not.
And such mechanisms have evolved.
So how does this argue? But if the evolution of neurons came from the need to outrun your conspecific, then that's different for the case where you're working as a
Yeah, and it's different for the case if, you know,
so
like.
do you still get the same spike? Would you still predict the evolution of neurons? For
I
the
don't know, I haven't looked at that, but it's, you know, as Malcolm pointed out, there were these Cloudina, which, as far as we know, never had nervous systems. Their solution to this problem was to develop shells. That's their solution. So you don't have to, this is not, you know, it's once you decide to be agile, this is actually why I like Nielsen's trachea theory, because that theory says that the first Neuralians were these pelagic plankton feeders that were feeding in the water column, right? And if you're feeding in the water column, developing a shell is going to work for you, right? But learning to get out of the way of predators is a good thing to do if you are a creature that has learned to live in the water column. So that's why I like that theory. But if you're on the seafloor, then, you know, just burrowing into the seafloor might be a good option. You don't want to run. And if you're burrowing into the seafloor, you don't need neurons.
So in Bayes' rule, you need so you have the probability of lambda given some event.
Yeah.
But you also just need the raw probability of lambda, right?
You need a prior,
Right,
but
the
the prior washes out almost immediately. Right? In sequential Bayesian inference, as long as as long as there isn't something actually upon me when I start this. In other words, when I pop out of the egg, and before my neurons connect together, something eats me, then I'm screwed. But if I can pop out of the egg and I've got 5 seconds to wire up my neurons, then whatever prior distribution there was is going to get washed out, and I have, now I'm confident there's nothing about to eat me. Right? So in sequential Bayesian inference, it really doesn't matter what your prior is. It goes away very quickly.
I have a question. So in neurons, we know that we have leaky channels. Do people have hypotheses before they kind of evolve to have that leaky channel? The cell. The animal before they evolve to use the neuron. What do they use before?
So if I understand you correctly, so these are actually really interesting, and I actually run a little computer simulation exercise with one of my undergraduate classes, showing that these things can behave optimally. They can distribute themselves across the seafloor in an optimal way without using any neurons, and their behavior, the way they steer themselves, is they drive themselves with cilia, so they're beating their cilia to move. and they have these little tufts on the back, and they spin, so they can go in a straight line. So as they swim, they're spinning along like this. And they just have a rule that says essentially if the environment is getting better, right, so if I'm getting into some more light conditions, into the kind of environment where there's likely to be lots of plankton for me to eat, then they just carry on. But if it starts to get kind of cold and dark, and this is not where a phytoplankton is likely to hang out, that little tuft on the back goes, and they flip in a random direction and carry on. So they just have this rule: if things are getting worse, I'm going to flip in a random direction, go some other direction. If things are getting better, I'll just keep on going where I am. And this is driven by sensors. So they have sensors around the ring here, which are just driving this thing to flip when these variables that are being monitored, whether those variables are going up or down, causing this thing to generate these little flips and head in the opposite direction. So you get this essentially gradient climbing behavior out of creatures that have no neurons. And plants can do this kind of thing. We tend not to think of plants as being very smart, but they are very smart. In fact, you could argue that they're smarter than us because they don't waste 20% of their metabolic energy on this crap. You know?
Chris. So if neurons evolved in response to predation, how does a predator choose prey without neurons?
The answer, we think, has to be that it had to start out as cannibalism. So we haven't done the simulations on this, but my favorite scenario is that it is Nielsen's trochea that are feeding in the water column, and they're getting bigger and bigger and bigger to the point where the big ones, like the zebrafish, the big trochea, suddenly decide, well, actually those little guys, being made of exactly the same stuff that I'm made of, are actually a better source of food than this other bullshit that I'm eating, right? Because the plankton in the water is trying to increase the costs for the predators. They're trying to make themselves unpalatable, which is what you see in the fossil record. But once one of these large trochea realizes, well, I could eat the little guys, it becomes a cannibalistic thing. And so you end up with, initially within one species, neurons evolving in both players as when they're small, an escape reflex, and when they're large, as an attack reflex. You see actually in the life cycle of these creatures that they are light-seeking when they're young. So they have a kind of behavioral flip like that these guys will head for the surface, find light for the first few days, and then they like it to be dark. So they'll go up to the surface and float along on the surface for a few days, and then they'll head down to the bottom. So I guess my scenario would be, it started as cannibalism. It was one species eating itself that drove the evolution of the first neurons.
All right, we're going to have to leave it at that. Let's thank the speaker. Thank you.